We engineer cell lines for cultivated meat and we sell these to other cultivated meat companies. I'll try and keep my own strong opinions at bay, which may be incorrect, but the scale system doesn't do texture and anything like that. It grows cells. It's more adept at growing high value, lowish volume cells. So things that might go into a cell therapy, things that might be used to develop a vaccine, things at that scale where you can either get multiple doses with relatively small production run. Cultivated meat needs to be at a ginormous scale. I'm talking like insanely high. And for this to happen, basically every bit of the supply chain needs to be working amazingly well.
Hi, Adam. How are you?
I'm good. How are you?
I'm good as well. And I'm so excited to talk to you, as it's obvious. But I'm going to start off with a basic question. I just want to know, what is it that you do at Unicorn Biotechnologies and how it's making a revolution in a domain which is so important and upcoming?
Okay, I'll start with why. Maybe I'll start with the why and then I'll move on to the how a little bit. So without giving you my life history, I did a PhD in cancer research and then I worked in stem cells. They're kind of opposite sides of the same coin, really. But I moved into I worked for a large industrial company after my PhD. And one of the problems was scaling any of this technology up. So it's brilliant doing sort of whatever we class as innovation in universities, but it's very then different in getting into the real world and actually affecting people. To see the value in the ideas, the rest of the world cares about execution. So they care about how you actually make that thing a reality. So I worked in, a couple of universities, a couple of large companies, a couple of startups, and then decided in my own insanity to start our own company. So what we focus on at Unicorn is really, how do you take bench scale research and how do you scale it up, or we scale it out? What tools do you need to do that? So I spent the better part of my life in a tissue culture hood for not the last 15 years, moving liquid between containers. I don't need a PhD to do that, nor does anyone else. I'm not degenerating the work that's done in the lab. I just think by this point, we should have automated some of this stuff. We didn't think there was brilliant solutions on the market. So given that I've experienced the pain firsthand in actually doing it, we thought we'd build something that would help automate and streamline cell culture workflows. So this is both for like R&D applications, but also for scale up. So this is anything ranging. We do some projects in cultivated meat, but the system isn't designed for that. But it's in projects ranging from sort of climate tech projects that use cells through to drug discovery, through to, pharmaceutical development, things like that. So the device in theory has, or it doesn't in theory because we're actually already selling it, but has impacts in getting basic research out of the lab and towards more of a sort of commercial impact, be that therapeutic or whatever. So yeah, I don't know if I've really answered the question, but that's the ethos behind it, and that's why we exist. And so if you want me to tell you more, then ask me more questions.
I wanted to know that your product Emmet, I understand that you've used AI and you've integrated AI and technology into how we can, like you said, automate the entire process. And a couple of questions that are coming into my mind. First of all, how much human intervention is still required in this entire process?
So I mean, at the moment, so it's always a work in progress. So at the moment, the device, or we give them a person. So it's named after. Well, maybe I shouldn't say this because then I'll get some corporation come after me. But it's named after Emmett and Mart. We've got another product that will come out eventually called Marty. These are Back to the Future characters. We thought so. They're
Right.
from well, we're nerds, so we like film references. We did think about Lord of the Rings and other things like that, but we decided on these ones. So we tried to make it like almost like a person. But the cell culture workflow, basically we've automated all the steps of it. So that's like you seed the culture, you then usually subculture the cells, you do different things with their stem cells, you differentiate them. So the system can handle all of this workflow, the same things that a human would do in a tissue culture hood, the machine does. But then the difference with like cell culture is it depends on the system. So I'm choosing my words carefully. If you're doing manual cell culture, you're process control is basically, does it look yellow, i.e. is it infected because there's phenol reds in the media and that the pH shifts and then the color changes. And then what you do is you look at it down a microscope and go, yeah, it looks about right. That's effectively process control for cell culture. And that's mad. For anyone that works in bioproduction, they're like, no. So we've kind of taken cell culture and then put our own, so we collect our own data sets in line with this, different data sets. And then the machine learning comes in theory. So machine learning is applied statistics. So you basically take that data off and you say, okay, given these set of circumstances, what's the ideal, what should
Okay.
ideally happen? And then you feed that back into the beginning of the process. So
Uh-huh.
yes, we do use machine learning to work out what a human might not notice. Really
Uh-huh.
experienced humans are really good at it. So they'll look at it and go, this isn't working because it doesn't look right and things like that. But that's
Uh-huh.
not really very scalable. So that's where we've already physically automated the process using a machine-driven process, but we're still doing the feedback loop at the moment where we basically take data, train models, and then use those models to work out a better way to do it. So the ultimate goal really is that this is the kind of highfalutin vision, is a kind of chat GPT-esque. You ask the system, please make cardiomyocytes. How do I do that? The system tells you how to do that. And then not only does that, it optimizes it for you in a quicker feedback loop than you as a human could probably work out.
Okay. But then, like you spoke about the data sets. So what have you basically trained it on? Because I'm guessing that it's going to evolve, like everything that we're
Yeah,
doing.
so it's things like imaging data, flow cytometry data, what else? Other data sets, sort of standard metabolic sensors, some other
Uh-huh.
cool projects that I can't talk about publicly, but it's sets of biological data that in an ideal world, we do one of two things. We either use complicated data. So let's say one thing, we could use next generation sequencing and say, right, we're going to do an experiment where we do loads of NGS, and then we're going to work out how we can predict the best outcome. The problem with that is it's not really very in lines, or you'd take the samples off and you ship them somewhere else. Then somebody does the NGS data and then you send it back. That isn't good for like a learning process because you want it to be fast. So we do things like that, but then we also build other systems that we integrate with the device that then
Uh-huh.
measure something in a more kind of real-time way. So that what you get is like we work out the process happens, you get data, but you get the data instantly, and then you can feed that into machine learning models straight away. And then over time, you build up a huge repository of data that basically says, with these processes, particularly anything around cells, we understand them so well from a data perspective, but we just then plug it into the machine, and the machine can then actually we want to get it to make its own decisions. So kind of like, it does something and goes, well, based on my experience, something's not quite right here. I should try something else. A bit like a human would, but just faster because you're doing it.
Mm.
You're doing this 24/7. So the device operates 24 hours a day. So
Right.
yeah, maybe it's a bit, for some people, it's a bit too, what's the word? 1984, or it's a bit too, you're removing human entirely. But I mean, if you look at it in a different way, no one complains. Well, we're having a conversation on the internet where there's data fed through cables and you get switching tables that decide which way the data goes in the packets of information. No one goes, oh my Lord, like my grandmother was a telephonist. So she used to do the cable, the routing for phone calls. No one ever goes, oh, I wish that still exists, because technology allows us to do other things.
Right.
So yeah, we're not trying to we're trying to like make the scientists or the people who are manufacturing make it. They work with the machine to basically
Uh-huh.
say, here's an experiment, let's design it, let's execute it, but let's not learn from the data as quickly as possible.
Okay. I now understand when you said that we have engineers and then we have people who are
Yeah.
scientists. I understand that.
So one of the problems I found when I've been working in places, I've worked in engineering sort of
Mhm.
heavy firms, and I've worked in science heavy firms. And science and engineers often don't speak the same language. The best example I'd give is actually from chemistry, not from engineering, but I've moonlighted as a lot of different things. So chemists, they call things a dry reaction. And when they say a dry reaction, I was like, how the hell do you get a dry reaction, like a chemistry reaction? And they just mean without water. So you can
Mhm.
have it in a different solvent, but the solvent isn't water. So between science and engineering, the way engineers talk about things isn't the way that scientists talk about things. And until you get people, and I know this maybe is cliched, but until you get them in the same room discussing with each other, I'll say is the polite way to put it, maybe arguing sometimes. The scientist doesn't really necessarily understand the engineer's world, and the engineer doesn't necessarily understand the scientist's world. I'll
Mhm.
give you an example from what we do. Engineers are used to workshops and they're used to things disorganized or organized chaos. So they're used to things like in a workshop environment, you can imagine it. It's got bits of metal, motors, pumps, stuff everywhere. Go into a science lab and something like this, everything's pretty clean and reasonably tight. Well, the clean is the main thing, sterility. So if you grow anything biological, if you get any bacteria in there, it's
Mhm.
game over. The bacteria multiply quicker than the mammalian cells. They consume all the resource and the mammalian cells die. So bringing scientists and engineers into the same space is very good to build interdisciplinary things. And that's been like a constant through my career. I've tried to become all kinds of different things, and now I just get to do that in the company every day.
Yeah, I actually wanted to ask you one more question that I'm sure that there must have been challenges. You've named one when you
Yeah,
were
yeah. So I don't know what challenges do you want to hear about? So
technology challenges.
technology challenges, not just life challenges. Okay. Yeah, getting people to speak the same language. Most of the people that we, not all, but most of the people that we end up coming into startups, usually, especially the science side, they come from academia.
Mhm.
Academia is all about papers and publications. Scientific publications are their currency. Industry is about a value to customer, and they're not necessarily the same alignment. So
Mhm.
showing something new and exciting, but you have only done it once or very limited amount of times is not the same as getting it to work. When we sell things to a customer, it has to work. It's like there's not a
Right.
debate about it. Whereas academia, let's say, operates at the kind of more novelty end.
Mhm.
We have to turn whatever we do novelty stuff as well because we patent things, but we have to turn that into a real world thing that works in a customer's
Yes.
hand reproducibly. And there's a different level of sort of almost tolerance there. Academia has much wider tolerance for error, basically, and industry is less so. And then if you add on like things like regulations, so things like if
Right.
you're a therapeutic developer, you need to comply with those regulations. Academia often has the freedom to not necessarily comply to the same degree. So that's another challenge. It's where people come from. So
Mhm.
whenever we hire people, I'll say this out loud. I don't care where you did your PhD. If you did a PhD, some of the people we've hired don't have PhDs because that doesn't matter. I care more about how you think and how you act. If you think and act like a startup, then or fast paced and try and work through things quickly, then you'll fit in here. And I think in any startup, to be honest. Whereas if you're wanting either unlimited freedom, even though academia you don't get unlimited freedom because your freedom is defined based on what the funding calls say they want you to work on, or if you want really structured processes and really like granular detail on what your job is, then you're probably better in a larger company. So some of the challenges are bringing in people, and then you bring in scientists and engineers together. I'm just talking about people. I'm not talking about tech now. What else about the tech? Sorry, I end up thinking about because actually it's everything. I'll quote one of my previous line managers. Everything is a people problem. Actually, it's a people
Mhm.
problem first and a technology problem second. I didn't used to believe this. It is. It is. It's always
Now that you're a
getting people
founder,
to
now you know.
yeah. Getting people to work together really effectively is really well. No, it's not hard. It's just people are people.
Mhm.
So what about technology? Yeah, it's things like engineers understanding what sterility means, and in the sense that they understand what it theoretically means, but they don't know what it means in practice. So some of the people that we've had work for us used to work in nuclear engineering, other forms of engineering, and they didn't need to consider sterility as part of the design process.
Yes.
Things like material chemistry. So cells, especially stem cells, they don't respond kindly to certain materials because they just don't like them because there's some, I don't know, it messes up the electron transport chain or it causes reactive oxygen stress or something like that. An engineer isn't trained to think about biology, it's trained to think about design constraints and specifications and how you build it. So when we've mixed those two worlds together, it's taken a little bit of time for people to understand where each other's coming from. The scientist won't understand that. You know, when you look at a product, an everyday life product, and you think, why is it not made like this? This makes so much more sense to be made a different way. 9 times out of 10, it's because of probably, there's a few reasons. It'll be material compliances. They just don't work together. It'll be the cost to manufacture would be so high, it would be economically unfeasible to build it. And probably 7 or 8 other real engineering reasons why they can't do that. But to the uninformed non-engineer, they'll just go, oh, that's just annoying. Whereas there's usually a reason or a regulatory constraint. The regulators say you must produce it in this way.
Yes.
Therefore, that influences the design of the engineering. That's another thing that, let's say, more R&D engineers don't think about. It's called design for manufacture. How do you design something that you can produce it as a mass-produced product at a cost that a consumer will buy? It's like, I don't know, TVs or something like that. If you had to pay 10 grand for a TV, or whatever amount of money, a high amount for whatever geography you're in, you would, you probably not. But they're only mass adopted when the price point reaches a certain price point. And you have to think about these things, and not everyone thinks about them. So it's kind of a mix of people, technology, design constraints, and then the science has to work somehow in amongst all this. We often joke that we're playing on hard mode. So we've taken engineering, which isn't easy, is not a trivial thing. If you look at all the products you use, there's usually some heavily, it doesn't matter what it is, there's some heavily involved engineering in that, and that's quite hard. And then you add in science, which is not necessarily the most reproducible thing in the world. So we're putting the two together. I find it a lot of fun, but yeah, that can be challenging.
That can be challenging as well. You touched upon the regulatory compliance, but I think before that, I'll backtrack a little just to understand more about what you're doing. So it's more like if I have an idea and I feed it to your system, and it will tell me a faster way to do it, or if it is possible or not. If it is possible, it may tell me ways to do it rather than me spending hours and hours in the laboratory and then finding out, oh, it doesn't work. So it cuts down that time and it helps me get to the actual conclusion very easily. But on the ground of it, when the tech helps get me to the solution, but the actual culture, the cell cultivation that has to happens via humans. Is my understanding correct?
So no.
AI comes there as well.
It's all via machine. So we may be very unusual that we cross too many domains. So we build physical hardware. So we build devices. I mean, it's always easier with pictures. We build the hardware. We also make the software ourselves, and we do the science inside it as well. So we do all, I don't know, I hate this term, I'm going to use it. We are a full stack company, whatever the hell that, we do all the levels. So currently we have sold hardware systems that have software on it, obviously, to other scientific institutions and companies around the world. The way they will usually work with us is they will send us, not always, but they'll send us material. We'll grow it in our system. We'll produce like data to suggest that works for their purpose. And then if that works, we sell them a system. So that's kind of what we do. The direction we're heading in. So at the moment is very user defined. So the user will say, I would like you to culture cells, and it's all schematic. Please execute this workflow for me. But the longer term vision is we're integrating the things that are happening in the machine learning world, or the LLM AI world. So, you know, people have got used to using ChatGPT now, and it's not great. It has problems, but that kind of human interaction is the direction we want to take. So this is where you start off. If you've got almost a system that the machine or the software helps you decide what to do, the machine helps you execute it and produce data, and the data gets fed back into the beginning of the process. So you've got a nice loop. So yeah, so the system at the moment is a hardware system that automates things, and it's moved towards a more smart system, if you like.
When you say that it produces data, that is the cell cultivation, or that is the
So but both it does it at the same time. Yeah. So it's doing the cell culture,
Mm-hmm.
generating data because we've got different, and then feeding it back. Yeah. Maybe I don't know whether I'm, it's just this is, so if you think about it differently, this is what humans do. So we either observe something or we think about something.
Mm-hmm.
And then we plan something, and then we use our eyes and hands to do something, and then
Mm-hmm.
we generally look at something and go, well, that's interesting, I wonder what that means, and then we go back. So that's kind of the scientific process. We're just removing the bit where the human goes with his arms and moves things about, and we do that with our system, but then we're also augmenting the sort of data side of it. So data is produced by systems, and then humans might look at them or analyze them, but then you want to feed that back into the loop quickly, like you would a child learning about something. So this is then where the machine learning and the data science comes in, if that
Okay.
makes sense.
No, it does. It
So
does
maybe
make
maybe it's too broad. We're unusual in that most people like pick up one thing.
right.
There'll be like a company that will say we're a data science company and we do LLMs for science, and that's interesting. But these tend to be, maybe I'm being too facetious to them. They're often wrappers of another. We don't develop our own foundational models at the moment. That's a bit too far for us. But they're often kind of user interface companies in ways. We know how to structure data. We know how to build hardware. We know how to create software. So we kind of just move the pieces of the puzzle about ourselves, and it's fun.
I think you're trying to refrain the using the word full stack,
Yeah,
but I
yeah.
I get the
I'm just sorry. I'm just redescribing, possibly in a very patronizing way.
no, but that's good. It's really helping me understand what you're doing. And like you said, that companies normally pick one out of the
Yeah.
two,
So
right?
there's a risk here. There's a risk. So I don't know. Maybe it's, I like to take the best bits from different things and mash them together. The
Mm-hmm.
risk here is that I don't know, by not, we do focus because we're focused on automation platforms, and to do these things we need to integrate it. But the risk here is, or the notional risk is, that you, by not focusing on one of the small angles, you don't become sort of, I don't know, world leader or expert in that thing, or be known for that thing. But the barrier I see all the time is with the AI type companies, is they go, ta-da, we've made a new model. And then the
Mm,
wet lab scientists go, well, that's great. How do I implement this in a wet lab?
yeah.
It's like people have sort of forgotten that we're not. So if you make biological material, or you're making it to make a vaccine or whatever it is, you're not dealing in bits and bytes. You're dealing in atoms and carbon.
Yes.
You need to find a way to combine them together and make the thing. It's
Right.
not like we can
It should
just
not
chuck it
be
on.
theory.
Yeah, yeah. The modeling has, like, if you look at like AlphaFold and things like that, is kind of crazy. This is biophysics, is beyond my mental capabilities, but, and that's all useful. They're great tools. But okay, AlphaFolds comes up with a prediction of something, and you go, right, we can make a therapeutic now. Okay, well, there you go, chemists, over to you, or whoever it is. They then have to make the thing that then
Mm-hmm.
becomes the drug or whatever it is.
Right.
I want to be involved in the making of the thing, because if you make the thing, then that's where you then, I don't know, let's insulin or whatever molecules or whatever things you're trying to make, be it cell therapies, vaccines, small molecules, whatever it is. If it then takes, I don't know, 10 years to make the thing, great. The
Mm-hmm.
AI sped us up by like a month, like in the
Right.
grand scheme of things. So you've got to have some sort of integration between all these fancy machine learning models and hardware. Because if you don't
Right.
have that, then it doesn't work for me. Or hardware, robotics, automation, however which way you want to call it.
So I think, Adam, when we talk about cell cultivation and the machine is learning, but I'm sure that there must be some rounds of inaccuracies, or let's
Yeah, yeah.
say, yeah. So then, how does your system help, or like maybe combat that? Because I'm
So
sure that like the first patch is good, and maybe like the next, like, have you faced that problem?
So we probably haven't existed for long enough to face the problem yet, is more the answer. So the answer is no, we haven't really,
Okay.
because we are predominantly with the system. It's taking protocols that are in the public domain through publications and things like that. So
Mm-hmm.
Some published research doesn't actually work, and we all know that. But if it's published and it works, then it generally works pretty quickly. And then basically the system's advantage is that instead of you using human labor to produce it, you use machine labor, and you
Hmm.
can go off and do something else. So we haven't experienced it too much, but we're only still at the beginning of feeding the data background in a circle. The
Okay.
argument I'd sort of put back the other way is that humans are also incredibly, at least when I don't like the word untrained, they're less trained. So if you're, say, a junior scientist that's just figuring out, like you'll read a protocol and you go, I don't really know why I'm doing half of these steps, but I'll do it because I know I need to do it. And then you forget to do a bit, or you decide to take a shortcut, or something happens and you need to make an adjustment. And you make the adjustment and you go, oh crap, oh dear, that did not work.
Hmm.
So humans make a lot of mistakes to do this anyway, as well. So yeah, eventually, as you start to train models, machines will make mistakes. There is a
Hmm.
theory, though, that they should, in theory, on who you are, it should learn pretty fast. It should go, I did this thing and it didn't work. So I shouldn't do that thing again. And it'll try and work. The goal is to try and work out what it did wrong itself in by using the data that it can monitor. I'll give you an example of, like, I don't know, in our system, we have a gimbal in it, which is a bit like a gimbal in photography. So it levels itself out within zero point 0.1 of a degree. A human can't even tell what the difference of zero point 0.1 of a degree is, but the machine can record that data and they might notice, oh, it was zero point 0.3 of a degree. I'm just sort of speculating here. I'm just making up a
Hmm.
use case.
Yeah.
And the machine would know it, but the human wouldn't. The human wouldn't even have known that piece of data. Or take, I don't know, the temperature fluctuated for some reason and that affect the process. The human wouldn't know it. Or if you look at cell culture as a whole process, there's a whole load of things that we do wrong that we shouldn't do. We take media out of a fridge and we put it into a water bath to heat it up to 37 degrees. And we put it back in the fridge. And we do this over and over again. That's a way to thermally slightly decompose what's in media. But we do it
Hmm.
and it kind of works, but it also kind of doesn't, because then the reproducibility between labs goes out the window. But that's just one example. Whereas if you have a system that controls the temperature much more precisely, then you remove you minimize that variable's impact. And that's the reason for machines. So that's a long-winded answer to say we haven't really. Well, there's the system. The errors the systems makes at the moment is more to do with tolerances in engineering. It's less to do with mistakes of thinking. But we're only at the beginning of, like, trying to make the machine think and process, not using the analogy of human, use the data to feed back through. We're only we've only been doing this for the last, I don't know, 4 or 5 months, things like that.
Okay. All right. And that's interesting to know that you've been doing it for a while, because then it also tells us that there's promise in the system.
Well, I think there is, but I'm biased. So I like, there's, so from us, there's promise in the system in that just automating the process is useful.
Mm.
Like, because it isn't there's always competitors on a market. There are other systems that do it that have disadvantages and advantages. The one major disadvantage we see is cost, because cost and
Yes.
size. They're usually very expensive, and they're usually the size of a house, and houses usually are expensive. So they're quite big, and we want to get them smaller and more modular,
Mhm.
so that you can
Mhm.
get the theory here is you get one and you go, well, that worked. I need more. You just get more devices as a copy and paste mechanism. And this then de-risks people financially getting into these markets, because somebody would have to come along and say, you've got a really high capex outlay. So the way we've built it is both kind of thinking about economics, thinking about science, and thinking about impact. So
What I was getting at is that during the cultivation, the AI is trained as per the data set that you have, in order, like, how it's supposed to maintain the oxygen, the humidity, the temperature and the pH and everything. And I'm sure that it's learning over time, right? It's learning over time, learning from mistakes. But if you had to judge the accuracy of how well it's doing, do you have some stats or something like that that can just help us understand how well the technology is able to perform as compared to what's already there in the market? I think what I'm trying to ask over here is that I understand the edge that unicorn biotechnologies has. It's given you everything in one go. But other than that, like, are there stats that could maybe help, benchmark what you're doing as compared to, let's say, what competitors are doing?
I don't
Anything
know.
on?
Off the top of my head, I don't have any like date. Like I'm thinking about what data do I have in my head. So maybe I'll answer the question in a roundabout way. So the one way we do it is we basically say, right, okay, these are what everyone expects, the benchmark of what it operates to. So if you talk about the pH needs to be this, or the dissolved
Mm-hmm.
oxygen needs to be this, and then we can make a device that maintains that process with a 0.1% accuracy. That
Right.
doesn't really tell you, if you leave the system to its own devices, just to experiment almost. I don't know how I'd benchmark that against other systems. The reason it'd be difficult as well is most of these kind of almost feedback loop systems, they don't exist for mammalian cells,
Right.
particularly. There's people that make digital twins and they kind of do process control, but it's more to do with like, this is more like fermentation or
Mm-hmm.
like more microbiology applications where basically the system will say, I don't know, the glucose has been depleted, and then they basically just dump a load of glucose back in. So there's systems that do that. Maybe I'm not aware of them. Maybe I'm blissfully ignorantly aware of other systems that do this for mammalian cells, but I haven't really seen them. Generally, it's either you look at, you've got a big, I don't know, stir tank reactor or something like that, and it monitors the pH over time, and the
Right.
lactate goes up, the glucose goes down. Classic, organisms consume carbon sources in the form of glucose often, and then they kick out lactate as a waste product. So they monitor those things, but that doesn't really do anything in the system. It's just more things like, oh, we've noticed that this has gone wrong, or it's a really simple feedback loop. But for mammalian systems, at least for normal culture, so adherent the culture we do in the lab, no one even
Mm-hmm.
looks at that right now, like at all. Like a bench scientist in an R&D lab doesn't, again, and going back to my earlier comment, they'll look at the pH indicator and they'll look at the phenol red color and they'll go, oh, it's gone a bit yellow. That's probably not a good thing. There's no
Mm-hmm.
like, the measurement is the human eye. There's no like digitized data. So
Okay.
the answer to that question is I'd have to think about how I would compare the learning ability of our system versus other systems, because I don't think any other systems really learn from their data, really. It's
Right.
more, it's more like like the thing they do with the data, it's more like QC. It's like a quality control. It says this batch reached this threshold for whatever number you want to use. It's more like a quality control metric.
Uh-huh.
And the data goes in a file somewhere in a report. And then if the
Mm-hmm.
regulators ever come back around and something went wrong with that batch, and they'll say, "That was all your process, your
Yeah.
CMC, and things like that." So it's used more for a weirdly, like not litigative, because it's not litigation, but it's used more for, quality control. Never understood why, like when I worked for this large company, I won't name them in case there's some trouble against it. We collected loads of data, didn't do anything with it. And we seem to store it all, and
Mm-hmm.
not really use it. And it's weird. It's a weird thing where, or the data was used just on a per experiment basis to reach a threshold. So you'd say, I don't know, for manufacturing, we need it to be 30% pure or whatever purity it was. And then it would
Mm-hmm.
then be like, oh, well, the batch is passed. And then you just do this over and over again. But there's not so much, I was trying to do this in this company, was do trend analysis and try to do some data science to try and this is where I started getting into all this, was years ago. So yeah, I don't think I've answered your question, but I don't think I can. I don't know what data I would produce that would be able to
Yes,
somehow
I guess
benchmark.
what I'm trying to ask here. I'm sorry, I cut you in between.
No,
Uh,
carry on.
what I'm trying to ask is, like, there are companies who are trying themselves to have an in-house AI database which they can leverage, and they keep failing. And then they reach out to other companies, like maybe, like you said, a science-backed company would be reaching out to engineers that, can you do this? And they'll be like, no, we need time, or there are these errors. So maybe since you are able to do it, you're able to bring both the the science and the engineering together, and you've been able to produce a product which is able to perform at a certain level. Maybe like when I meant stats, what I meant was that anything that can tell us that, hey, you were not able to do this, we are able to do this with maybe this much accuracy, or maybe the response time was correct and accurate by this much percent. Like that's where I was trying to um, gauge
Okay, I see.
who you are.
Yeah, yeah. So I again, these are all like work in progress. At the moment,
Right.
the core value the system provides to the customers we have is just it. Actually, in the first instance, is just automation. It's just
Mm-hmm.
basically having a machine do something that generally a human would tend to do. And the other
Mm-hmm.
advantage is that it's because it's like the bench scale process. The other problem we try to attack is, so if you go most um, a lot of suspension systems, particularly, we're all sort of, they're all kind of offshoots of fermentation systems. And then what's happened over time is people have gone, oh, it doesn't work. And then we're going to have to change the engineering a bit and put a stirrer in or do something else. So one of the problems then you have to, biology is a living entity, so it has to adapt to its new environment. So one of the problems is you take bench science and you go, right, well, this is great. We've got a potential product here or thing we could sell, but it's like we can only make like grams of it, so, or whatever, wet weight or whatever. So then what you do is you go, okay, I need a scale up system, and then you go and put it in a scale up system, and the biology just goes, I'm really sorry, but no, I don't like it here. I got used to down here and I'm now in here and I hate it. So then the process development scientist spends the next 7 to 10 years on average. That's the average time to develop, to then change the basic research process to fit it into the new engineering process. So the things that we do just means that the bench scientist just doesn't need to do this. They just need to put it into our
Right.
system. It operates in a similar enough way to the process they currently run that there's no need for process optimization particularly. So it's those two things, and the sort of the AI machine learning data side is the thing that I just don't know. Maybe there's a danger here that maybe then I'm about like an academic. I feel that this is something that's it's going to happen, and then
Yes.
it's just a case of who might win in
Within
that
certain.
space, so to
Yeah.
speak. So that's why we sell hardware. We help our customers that way, and then the AI end of it is a bit more sort of future thinking. Like if all this is true, where do we want to be in another couple years, whatever number of time? That's that side, if that makes sense.
Okay, that's good to know, and also gives me a little bit of perspective. I'll segue into something that I'm a little more interested in terms of the cultured meat, because I
Okay.
have seen like a lot of companies which are coming with plant-based meat, and they're there, but they're not as scalable. The science behind it, I just want to know from you, when they say that we've made this meat from plant, is it that what I understand is like in a stirred bioreactor, they put in all these things together, it keeps stirring, it keeps cultivating and growing, and once it's done, the mass is produced, it just more like a sieve, it takes it out, and here you go, this is the produce. Is that so when it comes to your technology? Do you get that mass like, or is it like you're able to produce the marbling effect as well and give it that texture and look?
No. Well, cultivated meat's a different thing. So we on the other side of the business that was involved in cultivated meat, we engineer cell lines for cultivated meat, and we sell these to other cultivated meat companies.
Okay.
I have my own, I'll try and keep my own strong opinions at bay, which may be incorrect, but the scale system doesn't do texture and like that. It grows cells.
Okay.
You can it grow, but it's more adept at growing high value, lowish volume cells. So things that might go into a cell therapy, things that might be used to develop a vaccine, things at that scale
Okay.
where you can either get multiple doses with a relatively small production run. Cultivated meat needs to be at a ginormous scale. I'm talking like
Yes.
insanely high. And for this to happen, basically every bit of the supply chain needs to be working amazingly well. So
Uh-huh.
you need to have very, very, very cheap media to grow the stuff in. You need to have cells that grow as fast as humanly possible. It's actually an interesting thing that I've been trying to work out is what is the biophysical limit of cell division? Like how, because basically, how quickly can the mass of a cell reproduce itself?
Mhm.
You want that number to be as low as possible because otherwise it's just not feasible. And then everything else needs to be very cheap, and they're kind of taking biopharmaceutical processes and trying to make them cheaper. It might happen, and they're when people say we've produced this,
Yeah.
I mean it's not usually it's enough for maybe an investor dinner and things like that, but it's not enough to feed any noticeable amount of people. Like in terms of like it, I mean, maybe the other companies would disagree. Maybe there's some inside knowledge I'm not aware of, but you have to make. There's a few companies, I won't mention them, I think are doing really good work in this space. I think they're considering the techno-economics, whereas I think scientists, I'm a scientist as well, sometimes kind of go, oh, I'll ignore the economics for now and we'll just work on the science problem, and the
Mhm.
economics will work itself out later. But I think there's some companies that have really thought through how are they going to get this like onto supermarket shelves. And then what usually happens is, so you grow the cultured cells as it like you try and grow them as efficiently possible, as cheaply as possible, and it's more like a mass of cellular material, which you
Okay.
then most of them are blending with other sources of protein, usually plant protein, because
Mhm.
we already know how to process plant protein at high volumes, because of the alternative protein space. And it's
Mhm.
basically then, hopefully, what you're hoping is that in the long term, that you can figure out how to reduce the costs of all the other parts of the supply chain. And then you may move away from plant protein. Or there's other companies that are putting in sort of the flavor components of it. That's one of the things we started off looking at, is thinking about what one of my background was making adipocytes, which are fat cells. So could you make the fat and just add the fats in, because often the fats will come, that's where the flavor comes from. But we moved away from that because we felt there was too many entrants into the space and thought there's people that have raised a lot of capital here, and it's called pulling the oxygen out of the room. Once a certain amount of capital has been raised, there's sometimes a risk that you come along and go, and the first thing you get when you go in an investor call is like such and such a company. And you're like, no, but we're different. Oh, but they've already got this in their in their mind, they see market leaders, and then it's harder for you
Right.
to raise investment. So yeah, I think that, and like, our system has nothing. The system isn't designed for cultivated meat. It'll produce maybe certain components that might go into it, maybe, but it's not gonna wholesale. It's useful for the R&D process, but it's not so much useful for the scale up, because the volumes you're talking are ginormous.
Insane.
They're huge. So yeah.
So if I have to take your personal opinion, it would be like if a company is stating that, I should probably ask them about
Yeah.
I
Well,
should
you
probably look into
my question usually to them is how much does it cost you to actually make this thing?
right,
And meat manufacturers thinking kilograms, not grams, like kilograms and tons.
right,
No one, as far as I'm aware, is producing that much. And some of the methods they're using for some of the companies are kind of insane. I know what they're like in the lab, and I just don't see how this is techno-economically feasible today, right now. But yeah, so I think maybe there's certain niches, especially like I think there's certain people that are, I'm trying not to say, there's certain people that aren't focused on, they're focused on pet food, or they're focused on a small sliver of like a high value market, a high value product, or perceived
mhm,
high value, where consumers are willing to spend more on those like meats, for example. Whereas if you're trying to do mass produced food where people they want it in volume and they want it cheap or relatively cheap. And cellular agriculture, cultivated meat, whatever you want to call it, is still, I'd say, at a reasonably early stage. But that doesn't mean that it can't get to a stage where the costs have come down, or you
mhm,
find ways to grow the material that don't require some of the expensive bits. And I know people working on that space, especially in the media space. So
mhm,
trying to reduce the costs of the media, because the media is a really expensive part of the process. But then you've got, as well, on top of this as well, you've got capex, people, opex,
right.
things like that. So yeah, I'd be asking like, how much does it actually cost you to produce this today? What's your projection into the future? And what's that based upon? Because
Mm-hmm.
there's some reports, and I won't name which reports, but I think they're speculative. They're non-scientific. They're just wild. They just assume certain things are going to happen in the future and it's magically going to be okay. But there's not actually any basis. There's no rationale on what that's based on. They're just like,
Right.
in the future, or they'll do orthogonal things. They'll look at another industry. Computer chips is always an example where in the 60s it was mentally expensive to produce a computer chip or per computer chip, and now it's a lot cheaper. So they just say, well, it happened in that industry, so it'll happen in this one, and that's fine from a social science perspective, but it's not a scientific way to look at things. So yeah, that's what I'd be asking.
Okay. I've got two questions. One is on the regulation. So when you're making or cultivating a particular cell of a certain type, how do you ensure that what you're producing is regulatory compliant? And then
Okay.
I'll go with that question first. Yes.
So in terms of regulatory compliant, I go to meetings where regulators are present and people discuss about evolving regulations. The
Okay.
general way to do it is, I mean, it's for like we have a quality management system. We record all of our data.
Uh-huh.
We make sure we source things in ways that we are pretty sure the regulators would like. So
Uh-huh.
it's usually if it's if it's cultivated meat, it's like veterinary certificates, things like this. So it's an
Right.
information collection exercise, really. So
Uh-huh.
we have to try and keep up with what the regulators want to they're wanting to see. But
Uh-huh.
to some extent, it's like we think about what are the best ways we can do this. That means that we're trying to think of like downstream impacts, negative impacts, basically, of doing it the way we're doing it. So that's the best that we can do. But because we're not a cultivated meat, there's no part of our business model that wants to be a cultivated meat manufacturer, and we are more supplying tools into that space. So
Uh-huh.
I like tech and tools, and so we're more of a tool company. So the regulations we have to adhere to are more to do with the tech and the tools. So it's
Okay.
things like CE marking, ISOs. So for the device, we stick to those. There's already reasonably good regulation, whereas cultivated meat is a novel food product. So I sometimes go to the regulatory forums and listen to what people are saying, but this changes quite frequently. So you've got to
Yes.
try and keep up with it. It's quite hard.
Okay. The second question I wanted to ask you is that when you go for meetings, like you said that I go into meetings and I have to like pitch like investors. So what would you say is like the winning point for you? I understand, yes, you've told me everything about how you're able to do it, and it's like it's a complete cycle. But if there's something that you would say that this is something that we've been able to do, for example, let's say the cost. Let's say there's a university and there these couple of 3, 4 guys, they've been able to produce something and now they want to scale up. And let's say if they come to you, so what advantage would they get when they come to you? Is do they get an advantage of maybe like this cost is low or anything like that?
Well, it depends on this. Then
What?
depends. So you said investors, customers. Investors and customers usually want different things. Investors want to see how you're going to be a path to a very valuable company. So the argument we make to investors is, well, my argument is, I'm a little bit biased, but that's what I believe. If we are connected on Earth, every living thing on Earth is connected by cellular biology. Everything.
Right.
You're alive. You have cells in you. If we can produce technologies that are cheaper, faster, more reliable, and learn themselves, there isn't a world in which this isn't valuable. It has to be valuable. And the market sizes are so the market sizes of the bioscience labor market is in the trillions of dollars. We're not talking billions, we're talking trillions. So if we
Hmm.
can automate and augment science, then we're in a valuable market. So investors care that you're in a valuable market. And then we just have a plan to try and capture that. It sounds horrible, capture that value as a company. So that's what we'd say to investors: huge potential, huge market, and here's the technology we're building. And it's also a problem because I also show an image of a computer from 19th. I've just said the computer analogy is dead, and now I'm going to use a computer analogy. I've got a picture of a computer from 1954, and then I've got compute like what the lab and now what it looks like today. It's come a long way. Labs look the same. About the 1950s, they don't, the way we do science hasn't really changed very much in, quite a long time. So that's why I say to investors. To customers, yeah, it depends on who you're talking to. For customers, we reckon we can probably get the cost down by 10 to what their process probably does. So that's usually what we say if it's a cost basis, but it's like the triangle of cost and quality. So
Mm-hmm.
we think we can get the cost down and either improve or keep the quality the same. Because often if you lower the cost, the quality goes down. For example, I don't know, you buy a washing machine and you get it half price of your old one, and they just put cheaper parts in it so it falls apart a week later. That's, yeah, you get it cheaper, but the quality's gone down. So it's not actually a win really, because you have to replace the washing machine again. So the arguments we make are usually a balance between cost and quality. So we
Right.
say it's as good as it's as good as a trained human,
Mm-hmm.
medium trained human, but it's not as expensive as a system that you need to, I don't know, build a building to put it in to even make the product. So they're the kind of arguments that we would use. But it really depends on the person. It depends on who we're talking to, because everyone has their different incentives within an
Right.
organization. Everyone has a different incentive. It depends on it depends on who you're talking to, really, if that makes
So that
sense.
means you've been able to, as compared to what's already there out in the market, you've been able to bring the cost down.
Yeah, it's like an ROI calculation to do with, or
Mm-hmm.
let's use different example industry, solar. You buy some solar panels and you'll go, okay, the solar panels cost me this much. How
Mm-hmm.
long is it before I get a return on investment in this? So we will say the machine will cost you this much, but if it's operating 24/7, these cost margins, you can expect break even point by this point, and you can expect like improve productivity by this point. So that's how we view the problem. It's not, people shouldn't really view it as like a simple, this is cheaper than that, because you've got to look into the future about things as well. So that's how we talk to people about how we think about it. But they might think about something completely different, because they might say things like, well, I've got this much budget in this massively large company. How are you
Mm-hmm.
going to help me improve my budget? They don't care about it sounds weird. They don't necessarily care about the whole organization. They care about what their sliver of the organization does. And if they're in
Mm-hmm.
manufacturing, they're going to, yeah, manufacturers care about price. It's usually the number one thing. Whereas if you're in, I don't know, if you're in the regulatory audit office, you're going to care about regulation. Like, how is this stop me gonna like get sued, or like does this
Right.
provide data that I can put into my quality assurance and quality control reports and things like
Mm-hmm.
that. So I build up like a knowledge, or I build up a knowledge base. So for them, the value is different. It's on a different part of the system.
Mm-hmm.
Depends on who we're talking to.
Okay. I had asked the team, like some of the technical people who've interacted with companies who are trying to inculcate AI into their system and are failing. And one thing that they've said is, I'm going to read it out: models often fail to capture turbulence, multi-phase
Okay.
flows, or cell microenvironment heterogeneity accurately in digital twins. So what are you doing differently to minimize these problems?
That is an interesting question. So I'll start with a quote from a physicist I used to work with. He doesn't like never hit. Heterogeneity is just a word that biologists use for something they don't understand. It's meant as a both a targeted attack on cell biologists, and also it's
Mhm.
true. The best defense to this that I can make is collect as much data as you can so that you can know how variable the system is. But then the way that we're trying to prevent this is like, they'll
Yes.
probably say this, but I'm going to guess that the systems that they're using to collect this data is probably humans, and it's probably manual. So
Okay.
if you had 10 people and if you ask 10 people to run the same experiment with all the same reagents, you'll get heterogeneity. Because if you think about it, like when somebody pipettes a liquid, they do
Mhm.
it at a different velocity to the next person, and somebody else does another thing and does another thing. So all that variance adds up. So by building a piece of hardware that has a known tolerance, you should be able to bring down that. If there is an impact of that on heterogeneity. Now, the flip side of that is maybe heterogeneity is like chaos theory. is just a thing that happens when you try to grow biological systems out there outside their normal environment. And that's a science problem. Years of research might find a way to figure out for certain organisms how to minimize heterogeneity. So people will clone cells, but then cloning leads to heterogeneity eventually
Mm-hmm.
because systems become disorganized. So I don't know if we're going to be able to combat that, but we can combat the engineering side of like, we're removing the variability of a human being, and even well-trained humans do things differently. You go into every single lab in the world, everyone does it. Like this protocol will say something, they all do it. They'll be like, people have come up with different ways of doing things, or they have different beliefs. Some of those beliefs are true, and some of them are like hearsay. Give you an example. Some people believe that shaking the plate before you put it in the incubator distributes the cells more evenly. Doesn't at all. People do it. It's like almost like they do it because somebody somewhere said you need to do this and they don't want to change it. But we've got like done some cool internal studies of just shaking plates and shaking them and see what happens to the cells. Yeah, if you do certain things like, I don't know, put them on a tilt, they'll all go to the bottom of a dish, of course, things like that. But this little shake that people do when they put stuff into an incubator does nothing, realistically. You're better off getting a spirit level and checking how level your incubator is. That's got probably a bigger effect.
Mm-hmm.
So people won't think about like the levelness of their floor if you care about that. And it's like this, but with everything. So it can be like, I don't know how often you take the media out of the fridge and all these things. None of that is recorded generally, none of it. And humans will do it differently to each. So by building a machine that does things with known tolerances in a known way, you should remove some of that heterogeneity. You should remove some of that heterogeneity, I think, I hope. That's the hypothesis.
You're able to do that?
Yeah. So we can test different things. The system has, we can test different. That's why you develop it, or the pumping rate and things like that. We control how fluid moves through the system with a known accuracy. A human uses a pipette and a manual
Yeah.
actuation system where they do this, and the speed at which stuff goes up and down is dependent on the human. And then you
Right.
learn as usually as a first year PhD or a master's student, and sometimes undergrad, if you put too much pressure on it, it blows the cells off the dish, for
Mm-hmm.
example. So there's all this kind of stuff. So the problem then is like, so a data scientist, if they don't go in the lab, won't see that. And they won't know the conditions of the data acquisition. They just see the result at the other end. And they'll go, why is there so much variability? Why is it so bad? And it can just be because human A and human B, human A has
Right.
been doing it for 20 years and knows. Human B has been doing it for the last 6 months and they're still figuring it out.
Mm-hmm.
So sometimes the data can be explained in that way, if that makes
All
sense.
right. And the last question, they had a question on, can some model be used for different type of cell culture? For example, mammalian cells, yeast cells, and bacterial cultures. I think you've answered this before. You pretty much
Maybe
everything.
they behave, if you can put like it depends. So this is a scientific argument. It depends on the cell type, the organism. Within mammalian cells, there's differences. So if you've got a stem cell and a cancer cell, they will have different growth rates. They will have different movement rate, locomotion rates. They'll have different
Mm-hmm.
because of intrinsic properties to the cell to do really grow them. Bacterial cells are very different in the way they divide. So are yeast. So the fundamental principles of how biology, say, I don't know, moves and divides might be similar, but the models would have to be different for each of these things. They're very unlikely to be the same, especially on kinetics. They're very unlikely to be the same models. You would have to. This is like trying to find a universal model, which is quite hard. Normally, what you would want to do, in my head, is you train the data on things that are as close as possible to that system. So that's how I would approach the problem. Maybe other physicists I know would say, yeah, we're going to find a universal model for this. I've got friends that work on stuff like this, but I'm a
Mm-hmm.
little bit more skeptical.
Okay. Thank you so much for your time, Adam. You've been
That's all right.
so right there with me with every question, and you've explained it so well. And I really enjoyed talking to you.
That's all right.